Hey, I think we'll get started. I think more people might be joining as we progress, but just want to start out by saying hello to everyone and thank you for attending this webinar on the Princess Margaret Living Biobank. My name is Cameron Orban, and I am the manager of the UHN Research and Innovation Core's Operations Office. My teams work with the core facilities in a number of ways to ensure their long-term sustainability. Through this webinar, we're focusing on promoting our core facilities that provide access to instrumentation and expertise to help you reach your research goals. UHN Research has several research core facilities that are available for booking by UHN and community researchers. In addition to the Princess Margaret Living Biobank, we have cores that specialize in, for example, flow cytometry, preclinical in vivo imaging, and single-cell genomics analysis. You can check out our website for more details. The PM Living Biobank at the Princess Margaret Cancer Center is a collaboration between PM researchers and UHN Biospecimen Core to establish a central repository and provide services for the use of patient-derived tumor organoids, or PDOs, and xenografts, which are PDX models. Our first presenter for today's webinar is Nguyen Pham. She's a staff scientist at the PM Living Biobank. Nguyen's academic path has revolved around a thorough exploration of the intricate workings of anti-cancer drugs and drug resistance, involving the exploration of various cancer models, including leukemia, colon, pancreatic, and lung cancer. One of her notable accomplishments is her pivotal role in establishing the PM Living Biobank, a valuable resource facilitating the creation and use of patient-derived tumor models across various cancer programs with PM investigators. Our second speaker is Nicolina Riccopinas. Sorry, I just massacred your last name, Nicolina. A staff scientist at the Princess Margaret Living Biobank core facility. With over 20 years of experience at the Princess Margaret Cancer Center, she has become a prominent figure in patient-derived organoids, those are PDOs. And Nicolina and her team cultivate PDOs, replicate cancer in the lab, and make those models accessible to researchers worldwide. Her leadership at Princess Margaret Living Biobank has been pivotal in expanding the scope and impact of research at UHM. Nicolina also leads multiple grant funding research projects and leverage organoid technologies in collaboration with various Princess Margaret researchers, further advancing innovation in cancer research. And now I'm pleased to turn things over to Nguyen, who will begin the webinar. I'll just, as a quick note, let you know that we are recording this webinar and it will be posted on the Research and Innovation Core's website and on the Princess Margaret Living Biobank website. Thanks so much.
Hello, can you all hear me?
Yes, now we can.
Okay, great. Thank you for the introduction, Cameron. My name is Nguyen, and Nikki and I would like to thank the Research Innovation Core support team for organizing this webinar. I will start with describing who and what the Living Biobank is all about, and we will describe the resources and expertise in the in vivo xenograft method, followed by Nikki describing the methods and studies for in vitro organoid patient-derived tumor models. Nikki will conclude with how we interact with researchers and industry partners. As you may know, UHN has existing biorepositories such as the PM Cancer Biobank to collect patient samples and primarily stored as snap frozen or formalin-fixed paraffin-embedded blocks for medical research. These are distributed for investigator-directed studies with approved review by Institution Ethics Review Board. Specimens are collected by conventional biobanks, tend to be limited in quantity, as shown here. Centimeters of specimens from the surgical collections, millimeters in size for the biopsies, and milliliter to liter of fluids. These could be quickly depleted by few studies. The Living Biobank is the next generation of biobanks. In collaboration with PM investigators, we have established patient-derived resources, PDO and PDX models, and provide these resources and expertise. to the research community. These methodologies are highly specialized and resource-intensive and time-consuming techniques. To generate tumor models which have high fidelity to their parent tumors, accumulating evidence shows potentially high clinical utility. As a consolidated resource and service core, we have effectively leveraged support from existing UHN cores, such as the Cancer Biobank to support the specimen storage, and digital teams for support in database tools to consolidate data and present to researchers the multidimensional data, including metadata, multi-omics, and quality control tests, and experimental data for users to visualize, query, and share data. As shown here, some of our data is shown on the cbioportal.ca, and also we have customized data showing our QC tests. Our activities are guided by an approved protocol reviewed by the UHN Research Ethics Protocol. And each collaborating investigator must provide a study-specific approved UHN REB protocol. Since 2016, we were formed and collaborate with PM researchers to consolidate models for the effective maintenance and distribution of resource to the research community. Whoops, sorry. Why do we need new patient-derived models? The alternative is to use simpler, long-time established monolayer cell cultures grown in plastic dishes, as shown in this left panel, which has some advantages and disadvantages. In comparison to the three-dimensional organoid models in a gel matrix and the in vivo xenograft system with tumor growth in the immunodeficient mouse host, these are progressively more complex biological systems to improve tumor modeling and functionally reflect closer patient tumors. Highlighted here are some of the PDX advantages in which they can mimic the parent tumor heterogeneity, the tumor microenvironment, and in some cases have higher explant success rate than in vitro models. Secondly, why do we need so many individual patient-derived models? What we learn from studying many types of cancers, as described in the Cancer Genome Atlas, one of the comprehensive cancer atlases, present the proteogenomics profiling of 11,000 patient tumors across 33 different tumor types, including 10 rare cancers. These results indicate that regardless of tumor types, individual patient tumors are unique and heterogeneous in their profiles. This suggests that we need tumor models other than monolayer cultures that better reflect this genetic complexity of individuals to evaluate functions of genes and the network that is relevant and impact on patient management and treatment outcomes. Could we study the molecular mechanisms of cell behavior using more complex PDX, PDO models? It is already known that standard monolayer models in preclinical drug testing fail to predict clinical outcomes over 80% of the time. Evidence is accumulating for the utility of these new patient-derived models, but more study is needed on larger number of models with consensus on standard operating protocols. I would like to share with you now some of our PDX methods and study designs. Generating a PDX resource is intensive with prolonged time to establish a stable model. The first cycle from patient collection specimen and implanted into the mouse generation, here we designate it as passage knot, could take up to 6 months, and serial transplanting fresh or cryopreserved donor tumor fragments for growth to a maximum humane size of 1.5 centimeter in largest diameter typically takes 2 to 4 months as you're cycling through from P0 to P1 and engrafting the fresh fragment from P1 to P2 and further expansions. Methodology for PDX establishment requires highly skilled staff for the surgical methods and to monitor and administer test agents. Histology is reviewed per mouse replicate, and DNA printing is performed routinely to verify the identity with the original tumor. Quality control tests include identifying the failed tumor growths due to lymphoma outgrowth, as you can see here in panel 2, with the slower growing tumor islands, or bacteria contaminated tumors occurring in the oral cancer specimens in panel 3. Models are tested regularly for murine pathogens, and each tumor cycle is divided and cryopreserved using an automated freezer for optimal cell viability. This slide shows a typical PDX drug efficacy study design. A cryotumor fragment is thawed and implanted into donor mice to grow as fresh donor fragment for the study phase. Each test condition is represented by 3 to 12 mouse replicates. Tumor growth is measured using a digital caliper, as shown here, for the length and width to calculate the tumor volume. This graph shows some days after tumor implant, with day 0 being the start of drug administration. The example here shows growth inhibition with a test anti-cancer agent. We have completed many such studies with different classes of anti-cancer agents. And our studies are performed using standard operating protocol with digital instruments, which data is read directly to a web-based licensed mouse database. We have been able to consolidate a rich data set and knowledge from our collaborating PM investigators. This enables us to review and improve on our methodologies and provide robust, standardized protocols for reliable and reproducible results. An example is our recent paper with Jessica Weiss, when she was a staff in the biostatistics core. Using PDX data, collected from studies over 3 years involving 25 models and 15 anti-cancer agents, we were able to address the question of how to optimize PDX study design. In this publication, we address how to improve study design by modeling and observing effects of various parameters. This included how to quantify tumor response, how many mouse replicates we should include, the number of measurements we should include, and the study duration. I would point to this publication for your interest. This list of our PDX inventory will give you the scope of our work with PM investigators as listed by tumor type, number of models established, and the collaborating research team. Most of this work has been supported through investigator-driven science and their grants and industry-sponsored collaborations. Additionally, since the inception of the Living Biobank, investigators have been able to secure projects that require the unique centralized resources and expertise of the Living Biobank. For example, we are participating in the Marathon of Hope Cancer Centers Network studies for the omics profiling of patient tumors with matched patient models. Second, we're also involved with the Ovarian Cancer Canada Consortium, as well as the pharmaceutical sponsors for biomarker-selected PDX efficacy studies on novel agents. These projects have helped to complete a more comprehensive molecular characterization of PDX, as well as added insight to their functional behavior. Additionally, together with investigators, the Living Biobank partakes in initiatives to support feasibility studies in new cancer types and method development including immune humanized mouse hosts for PDX studies. I would like to highlight that one of the strengths of the living biobank models is that there is an associated omics data set with these models. Like the tumor atlas presented in the earlier slide for patient tumors, we have our own local cbioportal.ca for patient level as well as patient-derived models, as shown here for some PDX, listed by the contributing investigators. And OncoPrint, and also highlighted here is in yellow, our newest data set for the organoid models. And OncoPrint groups lung PDX models by similar genetic abnormalities. For example, here we show some genes of interest. This allows us to evaluate targeted therapies in biomarker-selected studies. For example, if we selected KRAS mutant PDX to evaluate targeted therapies, would they all respond similarly? Such a study was performed by Josh, a PhD candidate in Dr. Child's lab. This study included the use of 10 KRAS mutant lung PDX models with 2 targeted agents, selinexor and trametinib. These 2 drugs target different proteins, which may impact on downstream signaling of KRAS tumors. The tumor growth curve here shows data from mouse replicates in control and treatment groups across all models. In the next panel, the data was summarized per model. Drugs show varying growth inhibition effects as a fraction compared to the vehicle-treated group, which may be dependent on specific KRAS mutations affecting specific amino acids or impacted by co-mutations. This work was resource-intensive, laborious, and time-consuming. In moving forwards, with some successes in optimizing growth conditions for the lung organoid platform, would it be possible to use a xenograft-derived organoid drug screen strategy as a more resource-effective platform for drug discovery screens? On that note, I would like to introduce Nikki to present the Living Biobank PDO platform.
Hi, everybody. Thank you, Nguyen, for the introduction. So my name is Nikki Radulovic, and I run the PDO organoid core, or patient-derived organoid core. And today I'll tell you a little bit about what we do, what organoids are, and how you can get to our models and how you can use them for your own research. So first, let's talk about the organoid technology very briefly. So organoids are self-organizing 3D cultures that are generated from stem cells or initiating cells that really give rise to the specific cell types of the tissue origin. The first adult stem organoid was reported in 2009, and it was generated from mouse LGR5 positive small intestine stem cells. And after this initial innovation, various organoids from different organs and stem cells have been generated from humans and other animals. There are 2 types of stem cells that could be used for organoid generation. There's pluripotent stem cells and adult stem cells. And now the organoid technology is utilized in different research fields and can be used as a model to study diseases, gene function, and cell development. So the key to growing organoids is really to give the right cues to the stem cells in the tissue to recreate the tissue structures. So you basically need some tissue niche factors that will stimulate pathways such as cell renewal and proliferation. So the stem cells will go on and differentiate into different types of cells that you can find in that tissue. It also needs a form of extracellular support matrix that contains your collagen, collagens, for example, that will support the structure of the organoids, this like sort of 3-dimension. So the way it works, you can isolate the tissue from normal and disease part. For example, here's an example of the colorectal adenocarcinoma. So you can take a piece of tissue from the normal epithelium and from a tumor epithelium. You cut up this tissue into very small fragments. You digest the tissue away to release the tumor cells and to get rid of all the connective proteins and tissues. And then you will embed those tumor cells in matrigel, which is really your extracellular support matrix. And then you put them at the bottom of the plate. Once it solidifies, then you can add the different media that contains these niche factors that will stimulate these stem cells in the tissue to actually start forming different structures. And organoid technology has wide applications, and we can use it for disease modeling. We can use them for drug testing. You can also study host microbiome interactions, and also you can biobank them to be used for research. Now, several groups have published generation of organoids from different tumor types, and some of them are shown here in the Cleaver's report. So efficiency, when it comes to efficiency, that really refers to success of generating organoids from tissues. However, the deficient efficiency is not really consistent in different papers. It can either be defined as organoid initiation, which is really any organoid growth after tissue processing. It can be defined as short-term cultures, which are really organoids that initially grow. You can expand them for a few passages, but they're not really renewable. We can't really cryopreserve them and have them later available for researchers. So when it comes to the living biobank, we are really working with long-term cultures. So these are the cultures that are renewable, so they can be expanded for at least 10 passages. And also there have been quality checked in our core. So the following is sort of the workflow that we follow from receiving the tissues to generating organoids to biobanking them. And you can kind of see that there's a large amount of data at each of these steps. So during pre-processing, we take the tissue. We also take a piece of the tissue for histology sample, and we also snap freeze the tissue so we can use it later to isolate the DNA or any other nucleic acids. We also, once we process the tissue, we record the total numbers of cells, the number of live cells, and the sample viability. And then we plate them for organoid generation, and we really use different media types depending on the tumor type. Sometimes we have multiple medias for that tumor type, and then we passage them. We record the passaging ratio, we take images, and the models that really make it more than 10 passages are those that we consider to biobank. So once we are happy with their growth, we then do a different kind of quality tests. We test them for bacteria, histology analysis. We also sometimes use flow cytometry to make sure that whatever is grown is actually epithelial cells or tumor cells. We can also implant them in in mice to make sure that they're actually tumoroids and they can generate tumors in mice. And we also have recently been putting all of the organoids that we generated through omics platforms. So we have whole exome sequencing, whole genome sequencing, RNA sequencing, and ATAC-seq sequencing available for some of these models. Then they're biobanked and they can be renewed. They can be, we also test the cryo regrowth in our core. So in PMLB core, we use various biospecimens to generate these organoids that include surgical tissues that are shown here on the left. Biopsy tissues is a really tiny biopsy here that's shown on the right. And we can also use fluids. So we have generated organoids from pleural fluids and generating from lung cancer patients, for example, and also ascites from ovarian cancer patients. So these tissues fluids are processed and the tumor cells are isolated. We plate them on in this extracellular matrix. And you can kind of see here that they usually just look like single cells after processing, but they generally organoid growth will appear anywhere between 1 to 2 weeks. There are many advantages of using 3D organoid cultures in research, and we have, for example, you can use them, there's patient-specific cultures. They resemble in vivo complexity of tissue architecture or the cell hierarchy. So you can see here some examples of pancreatic organoids. This is the phase contrast image, and this is an H&E. So you can see lumens are formation of lumens. Some of the organoids have single lumens, have multiple lumens. Some of them are very sort of cellular in structures, and that sort of recapitulates the tissue morphology. Also, the genomic complexity of patient tumors is preserved in these organoids, unlike 2D cell lines that really are where genetic changes occur quite a bit. Organoids are better for that. You can also culture them from small sample size. As I said, we can generate them from biopsies, and you can generate them from different regions from the same tumor, so you can kind of model the cancer heterogeneity. Organoids also mimic tissue cell hierarchy, so you can see a lot of differentiated types of cells in these organoids. So what I'm going to give you an example of lung organoids. So this is immunofluorescent slide images that shows the tissue. This is on the left-hand side is a mouse tissue of the lung airway. So epithelial cells in the lung airway will form cilia, and cilia cells are indicated here as red, which is basically stained for beta tubulin. On the right-hand side are the organoids that we've generated from lung patients. And you can also see, first of all, there's a polarity to these organoids, and you can see the cells that are expressing cilia. Another cell type that is found in lung organoids are mucinous cells. So here, this is an example of tissues, normal lung epithelium, and mucinous tumors. You can see the mucin cells are shown in green. And also, if we look in our organoids that are generated from lung, we can also see the presence of mucin-producing cells. So PMLB PDO models recapitulate tumor tissue architecture. As I said, this is more detailed slides for that. So you can see here the phase contrast, H&E, and immunohistochemistry state images of organoid models and original patient tissue. So organoids closely resemble the morphology and biomarker expression of tissue of origin. For example, organoid made from colorectal cancer. Here, patients show very polarized epithelium, lumen formations that are also present in the patient tissue. And these organoids are also positive for cytokeratin CK20 marker that's indicative of their colon epithelial origin. So all our organoids are analyzed by immunohistochemistry and histology. So organoids generated by PM Living Biobank come from various malignancies, including pancreatic, colorectal, lung, esophageal, ovarian, and breast. Here is the breakdown of the number of models that PMLB currently has for each tumor type. We work closely with various principal investigators who really guide our practices and development and support us on many levels. And again, a lot of these models are quality control approved, and this is our QC testing, a list of QC tests. Each deposited organoid model also comes with characterization sheet. So each sheet includes basic demographics of the patient, cancer diagnosis, and details on propagation for each organoid model. So if models are distributed to an outside lab, we also provide detailed SOPs, which include propagation, media recipe, freezing. We also have organoid training that's available. So if anybody wants to learn how to do this, or they are ready to receive one of our models, we always recommend that you come and work with us. And we have about 3-hour training programs where you teach how to grow them, cryopreserve them, and also revive them. Next, let's look at some applications of these models, and I'll talk about drug screening. So we have been using them to generate drug screening profiles for several PI-led projects. So we can perform organoid drug screening on any plate formats, including 384 and 1536 well plate. So organoids are, here is the example of typical drugging protocol. Organoids are dissociated to single cells, and then they're plated on multi-well plate, depending on which type of plate you need. Also, depending on the type of drug that's used, drugs are added either 1 day or 3 days post-plating, and then you measure the viability after a certain period of time. That can, we usually use basic ATP proliferation assay, but you can use microscopy, or you can use any other type of cell viability measurements on organoids. And I always say, whatever you can do, whatever you do with 2D lines or can do with 2D lines, you can also work with organoids in the same way. One of the first studies the PMLB participated in was the COMPASS study. So this was a large clinical study that was led by Drs. Jennifer Knox and Stephen Gallinger here at UHN. So they collected clinical and molecular data from patients with advanced pancreatic ductal adenocarcinoma. And you can see that generating PDOs was just one arm of the larger study. And the goal of PDO study was to generate and pharmacotype PDOs from patient biopsies and correlate therapy responses in patient PDO pairs. So whatever the drug the patient was getting, patient-derived organoid was treated with the same compound. So the standard chemotherapy sensitivity curves were generated for each organoid model. So these curves are showing dose response range with cell viability, and the results are summarized as area under the curve to evaluate ranges of drug sensitivity. So based on this, organoids models can be categorized in 3 groups: resistant, so these are the models that have that are shown here in red; intermediate, yellow, and sensitive would be the organoids that are indicated in blue color. So the way the drug sensitivity correlation works is basically, if the patient is sensitive to that drug in the clinic, we are looking for where this organoid sensitivity falls in. So if it correlates, the organoid should also fall within this category. Now, where can you find our models? So first, you can contact us directly to inquire about the models that are available for research. Also, a subset of PMLB models are currently visible on cancermodels.org database, and this also includes PDX and PDO models. So this is a database that is supported by the National Institutes of Health and National Cancer Institute, EMBL, and the Jackson Laboratory. So the database contains a list of models from institutions across North America and Europe. So it's a very useful database. Because in addition to our models, you can perhaps find other models that might be suitable for your own research. Our user community includes academic institutions and not-for-profit research centers. We have also worked with commercial entities such as pharmaceuticals and biotechnology groups. Our models have been used by many UHN investigators, resulting in a series of publications in high-impact journals. We have also collaborated with external academics, and our models were showcased in their publications. Now, what are the possible ways you can interact with us with organoid and PDX core? Well, first, we can help with model generation. So perhaps you might be interested in generating models from tumor types that are currently not in the biobank. You can contact either myself or Nguyen. We're always looking for tumor types to add to the biobank. So you might already have PDX models, for example, that you're interested in making models from, or organoid models, or depositing those in our core, so that we can do that as well. We offer grant support, so we can either give a letter of support, or you can include us as co-investigators. We can provide help with methodology and provide preliminary data even to help with the grant writing. We also provide SOPs if you are interested for various aspects of organoid and PDX work. So we are happy to share our protocols with our members, with UHN and external academics. And we also have in-house training, particularly for organoids, where we can teach you how to make them, we can teach you how to grow them, and maybe even help you with drug screening. We also have organoid consumables. So basically, we can provide organoid media for growth. We can also make conditioned media, such as Wnt and responding medias that are important in generating organoids for some of the tumor types. We can also provide quality assessment of existing models. Perhaps you already have some growing and you want to do histology, so we can provide some help. And also any other ideas are welcome. There's many other custom projects that are currently happening. So we are trying to find better ways to, for instance, incorporate CRISPR targeting in our models and so forth. Now, this brings me to the most important slide of the presentation, which are really acknowledgements. So PM Living Biobank is directed by Dr. Ming Chow and by the scientific advisory committees. These are the RPIs that are really help guide our development and our progress. I manage organoid work while Dr. Pham manages xenograft studies. We have a number of very talented technicians in both PDO and PDX cores who have been instrumental in supporting our work and in model development. And also we are supported by other UHN cores, including Animal Resource Center, PMC Biobank, Histobiomarker Lab, Cancer Registry, Genomics and Data Science, Research Ethics Boards, and Commercialization. So that would be it. Thank you for your attention, and we are happy to take any of your questions.
Yeah, I just want to start off by thanking both Nuan and Nicolina on this very informative webinar on the Princess Margaret Living Biobank and on both patient-derived xenografts and organoids. So yeah, before I begin the question session, I ask everyone to feel free to input any questions you have in the chat below, and we'll answer any questions that are posted. So the first question I see here is, how long does it take to establish an organoid model?
Okay, so that's a very good question. It really depends on the tumor type and on the biospecimen that we're actually using. So it can take anywhere from 1 to 4 months typically. We usually know by that time, but that means those model is completely deposited and it's passed all the quality assessments. So the bigger the tissue, the more cells we have, it's a higher chance that we can make organoid faster.
Great, thanks. And how much do these models cost?
So the next question in the chat is how much do these models cost?
Okay, so I guess I can start this. First of all, we don't really sell the models. We kind of work with all our models are distributed through MTAs, and they're only distributed to individuals or to labs that have ethically approved protocols, because in Canada, organoids and other type of patient models are actually still considered human tissue. So we distribute them using MTAs, and also, we do have processing fees to cover the costs of, growing these organoids and also, biobanking them. So I don't know if you have anything else to add to that, Nguyen.
Nope, I think you've covered that. The next question is about experience with PDO co-culture study with immune cells or fibroblasts.
Yeah, so currently when it comes to the living biobank core, we are, the organoids that we cryopreserve are really the epithelial component of, the tumor cells. Really, they are void of connective tissue. There's no immune cells that are in these models. But that being said, there's a lot of effort going on to make co-culture models, or these more complex models where you can layer on immune cells or fibroblasts. So there's a lot of PIs that are working on that. It's a bit more challenging, but the way that Living Biobank can support those efforts would be through providing the models and helping establish some of the protocols as well.
So the next question, I'll read it out myself, is concerning about commercial entities. How can they access PDX models? And is it only through subcontracting and in vivo study? We have many different contracts depending on what the commercial entities require, whether it be performing full service PDX, PDO studies in terms of drug efficacy, or the distribution of the models for the companies to use in their own R&D labs. So it is very much dependent on who we're working with that will have specific contracts. Nikki, any additions to that?
No, I think you've covered it all.
Okay. And the next question is for you.
Yes. Do you accept students from outside Canada to take the training for PDO? So we haven't had anybody from outside of Canada, but we have had people come in, or different researchers come from different institutes within Canada. So I think it would definitely be possible. I think you just have to maybe email me, and then we can discuss the logistics.
We have an example of that. Now we're actually making training videos to be provided to our collaborators if they cannot come on site. So we'll make specific customized training videos and sending it out. There is another question for you, Nikki, about organoids and contamination with bacteria and fungus.
Oh, okay. So typically when we're starting off and making the organoids, I think it depends on where the tissue comes from. So areas such as colon, esophagus, they're usually full of bacteria. So we do need to have a lot of we have different antibiotics that are used initially to suppress the growth of these, which includes primicin and other types of antibiotics, which can then later on can be scaled down as the model is growing. I would say once the model is already contaminated, it really depends on the level of contamination, and if you can actually clean it up, or if it's worth even cleaning up. So I think that that really depends again on the level of contamination. But your best bet is to initially make sure that the contamination doesn't happen. And we can, again, you can email me, and I can help you and provide some protocols for you.
The next question is, how can we access the models?
You wanna get to that, Nguyen?
Yeah, as Nikki mentioned, please email us directly because our inventory is updated real time. And sometimes if you view it through various interfaces, such as the data portal that we mentioned, the cancermodels.org, it may not have the real-time numbers. So depending on your needs, please email us directly. We have a PMLB email, or you can use our email. That would be best.
Okay, and then in terms of accessing, so also I guess I can add to that, is the way it works. We would help you identify the models of interest, whether or not that's through looking at the genetics or any other features that you need. And then you would, usually we have a sheet that you fill out to tell us what your project is about, how you're going to use these models. And then we use that information to, if it's an outside institute, then we get our MTA office to approve that, and then we work with you on distributing the models. And again, your protocol needs to be approved by the Research Ethic Board, either in UHN or if you're external by your own institute. There's one more question here, I think, that I should address possibly. It's about fresh tissue explants or patient-derived explant models, and if there's any interest. We haven't worked with those. I think that the issue with some of these things is the longevity of the models and how renewable they are. So as a biobank, we can really only biobank models that we can pass on to the next researcher. So I'm not really familiar with patient-derived explants models and what the longevity is, but I'm certainly open to suggestions and discussion. Is that it? There's no more questions? Yeah, I think that's it. I think we've covered them all.
I think there's one question that just came in. Someone posted, do we have info about gene mutation background for each sample?
Nguyen, you want to address that one?
So on the chat, there's a question about, do we have information about gene mutation background for each sample? We have the whole exome sequencing, and some of it would have some RNA sequencing data or gene panel sequencing. Not all of our models have genomics data, but some do, and we're willing to work with collaborators to continue to profile so that we complete the molecular profiling of our collection. This way we can prioritize. If the user demand is high, we definitely can continue profiling the models that are lacking molecular profiling. So we work with the collaborators to see what your needs are before we go off and do things without user input or scientific input.
Yeah. And I can add to that because, organoid core is sort of like a newer core, so we have less models than the PDX, right? So majority of our models, I would say at this point, probably at least 90% of our models do have been sequenced in the last year. So there should be the data available for mining. What medium mostly used for organoids? So that's another question. Again, every tumor type has a different media, and within that tumor type, we also use multiple medias. So there is always this base media that's kind of common for all the organoid medias, but it's quite tissue specific.
Please feel free to turn on your microphones and ask questions directly if you're not typing into the chat. We don't want to miss any questions, or feel free to email us, and we'll be happy to address your concerns.
I want to say thanks so much to both of you for this very interesting presentation. It looks like
Cameron, you're muted.
Oh, sorry.
No, he's not.
No,
I can
no,
hear
he's
him.
not.
I can hear him.
Oh, good. No, just thank you both very much. It was really interesting for all of us. It looks like we might be at an end of questions, so maybe we take the time now to say thank you for everyone's participation. And please do follow up directly with us if there are any questions that come to mind afterwards. And thanks again for the participation and for the great discussion. Thanks so much.
Thank you for having us.
Thanks, Noel.
Thank you.
Lena. Thanks, everyone.